It was a bright and sunny day when a hungry grasshopper decided to eat a soybean leaf. He was warned not to eat soybeans or any crops, but he wanted a delectable treat. Little did he realize what would happen to him and his RNA as he bit into that yummy leaf. As he digested it, his RNA was changed. The amount of eukaryotic genes expressed are controlled by the RNA interference or RNA silencing. RNA interference is a driving force among most eukaryotes, such as plants, animals, and fungi. Some plants have found a way to use RNA interference as a defense mechanism to strike back against those who dare to consume any part of it. This occurs when plants use RNA interference as the most reliable method to limit an invasion of foreign genes. The initiation of RNA interference is caused by small RNA molecules, such as microRNAs, miRNAs, or small interfering RNAs, siRNAs. These RNA molecules are contained within an RNA-inducing silencing complex, or RISC, which pairs small RNA molecules and proteins. Depending on the location of where RNA interference initiates, influences which mode of action to use. There are three modes of action for RNA interference. 1. Cleaving of mRNA. 2. Inhibiting translation. And 3. Transcriptional silencing. These modes of action can cause a genetic or developmental change within the chromatin structure during translation, determining a cell's fate and census, and can also cause cell death. Scientists are interested in transferring this defense mechanism to various plants that are vulnerable to pest invasions.
In the article, Crops That Kill Pests by Shutting Off Their Genes from Science Daily, there is new research being done to look at how we can manage pests through RNA interference in crops. We will explain the three modes of actions that the researchers could use when developing new pest management strategies. RNA modes of action. 1. Cleavage of RNA. The siRNA within the RISC pairs the messenger RNAs in order to cleave the mRNA exactly where the siRNA is bound. A protein referred to as the slicer is responsible for this cleavage. The mRNA fragments are then continuously cleaved by this process until it is completely degraded. The more siRNAs and miRNAs present, the faster an mRNA molecule can be broken down, which would lead to a low production of that specific protein. Steps. Double-stranded RNA is cleaved by the enzyme dicer. This produces small interfering RNAs. The siRNAs combine with proteins to make a RISC complex. RISC pairs with complementary sequences on mRNA. The complex then cleaves the mRNA multiple times using a slicer protein. Once cleavage is done, the RNA is degraded. 2. Inhibition of translation. Some miRNAs play a role in regulating genes by inhibiting the process of translation via their complementary mRNAs. Steps. Other regions of a double-stranded RNA molecule are cleaved by the enzyme dicer. This produces microRNAs. Some miRNAs combine with the protein within a RISC complex and pair imperfectly with an mRNA. This leads to the inhibition of translation in which these proteins cannot be produced at all.
3. Transcriptional silencing. Some siRNAs inhibit transcription by altering chromatin structure. These siRNAs bind with proteins to form a complex called a RNA-induced transcriptional silencer, or RITS. The siRNA region of a RITS is then combined to its complementary DNA sequence or an RNA molecule in the middle of being transcribed. There it represses transcription by attracting enzymes that methylate the ends of the histone proteins. Adding these methyl groups to histones causes them to bind to DNA even tighter, to the point where access to proteins and enzymes used for transcription is restricted. Steps. Other siRNAs attach to complementary sequences in DNA and attract methylating enzymes. These enzymes then methylate the DNA or histones, which causes the inhibition of transcription. In conclusion, RNA interference is a driving force amongst most eukaryotes. Certain plants have found a way to use RNA interference as a defense mechanism against hungry bugs. The initiation of RNA interference is caused by small RNA molecules, such as microRNAs or small interfering RNAs. When the RNA-induced silencing complex, or RISC, pairs with small RNA molecules and proteins, depending on the location of where RNA interference initiates, influences which mechanism to use. Three mechanisms for RNA interference are cleaving of mRNA, inhibiting translation and transcriptional silencing. These mechanisms can cause a genetic or developmental change in the chromatin structure during translation, determine a cell's fate and census, or cause cell death.
How will this affect pest management and agriculture?
Every farmer has the issues of insects in their field. This could really cut down on the use of insecticides on their corn and other crops, and also all the extra costs. We can use this in conjunction with or instead of BT corn. Instead of using insecticides, we can use RNA interference to alter the DNA of the insect. Pest management could be easier than ever once scientists and plant biotechnologists incorporate this into their crops.
And what do you think the consumer acceptance of this technology will be?
I think consumer acceptance of a new technology where we're not actually putting more pesticides on or inserting a toxin is going to be a lot higher than anything that we've ever had before, just because it's something that is the plant itself is doing, and we're not doing it ourselves. Like, we're not putting out the chemicals or anything like that. And we're not really putting out any toxins of any kind. We're just changing the DNA of the plant and the insects, and so it's possibly going to be a little bit different than the GMOs that we have today.